I'm sure now it is audible. So, I think the technical issue is already been sorted out. So, I think it is audible. So, I will repeat what I have just said before also. So, I think the Priyanka was asking about the scope of this course, and the scope of what I have tried to cover is many aspects related to genetic engineering, and the at the end the the purpose of this course is to give you a very glimpse of what is the potential of genetic engineering and what are the different protocols and procedures what you can do actually using this genetic engineering as a starting thing. And once you are fully aware of these aspects, I am sure you will develop some interest, as well as you are going to have some confidence in terms of when you go for when you would like to decide whether you would like to use the genetic engineering course as approach or genetic engineering field where the genetic engineering protocols are being used, and that actually will help you in deciding that future aspects. The second point is, suppose you go for would like to decide that okay, I would like to go to industry after completing my VTech or MTech, then we have actually discussed many technical aspects related to genetic engineering, and we have also discussed how to perform that in the laboratory as well. So, in that context, it is actually going to make you very confident when you go for a company job. For example, if I enter into a company and somebody will ask me, okay, you have to clone XYZ gene and you have to overexpress this protein for vaccine development or something like that, then many of the times you may have a theoretical knowledge that how to do that. But in this course we try to cover not only the theoretical knowledge but also the practical aspect as well. So when you go through with this course, you will become more confident. Definitely every lab or every institution is going to have its own way of setting up the things. So the minor detail may not be maybe something which you might have to learn in a some span of time or some time in your place, but the major thing you will be clear what need to be done next. So if I have to clone a protein, I would like to design a primer, that we have discussed, and we have also shown you how to design the primers, which I'm sure many of the textbooks never been discussed or never discuss these things actually. On the other hand, we have discussed about how to prepare the genomic library and cDNA library. So suppose if you face that kind of problems, then you can be able to handle that as well. Once you have done that, then you are going to have the purified or PCR product. That PCR product you can actually you can elute the PCR product from agarose gel, you can ligate it and then get the recombinant DNA. Then we have shown how to do the transformation also, we have shown how to prepare the competent cells as well. And so the idea of this course is to give you a complete information both at the theory as well as the practical aspect, so that it will make you more confident once you done with this course. The other thing what you were asking about is the important chapters for the exam. So in that regard, I have tried to cover all the aspects related to this course. So there is no such shortcuts, so that I can tell you that this aspect or this particular chapter is going to be more or less. So you might have to go through with the whole course. So and apart from that, the Priyanka is also asking about the delivery method utilizing virus as a DNA carrier. And if you recall, we have discussed about how the transaction is working and how the virus is infecting the mammalian cells. So you had the choices of different viruses like adenovirus, lentivirus, and so on. So you can actually produce the recombinant DNA, and then when you produce the viruses containing that recombinant DNA, that you can use to infect the new cells, and that's how you actually can use the virus as a DNA carrier. Now, if you have any more questions, maybe I will happy to answer. Otherwise, I will again continue with the some of the assignment questions, and we will going to discuss a few of those questions, and hopefully that itself is going to give you the answer of your queries, because I have chosen few questions based on my experience when I was discussing these questions in the discussion board itself. So many of the students were asking many things related to these questions, so I thought I should include those during this interaction sessions. So I'm sure now you might be I might be audible to you, and you can be able to post your questions. In the meantime, let's discuss the few more questions from the assignment 1 to 4. We are not going to discuss any question from the assignment 5 because it is still due by Wednesday. So let's wait for the next interaction sessions when we are going to discuss about the questions from the assignment 5 to assignment 8. So let's have a next questions. So the next questions. From the assignment 2, we have asked the question, what is the next net yield after completing oxidation of glucose molecule in glycolysis? And you can see here that we are going to show you the balance sheet, and the balance sheet is saying that you are actually, so if you see this question, we have given you the options in terms of the NADH as well as the ATP. So that actually is the same. So what you see is that in the step 1 to 4 you are actually using the 2 ATP molecule, and whereas in the subsequent step like figure step 6, step 7 and step 10, you are actually generating the NADH. So you are actually generating the 2 molecules of NADH and the 2 molecules of ATP. So in the step 6 of the glycolysis, you are generating the 2 molecule of NADH, and you are actually generating the 4 molecules of ATP from the step 7 and 8. So if you see that, what you are going to have is 2 NADH molecule and the 4 ATP. That is the total production. But if I subtract this 2 ATP which is being utilized, what I am getting is that the 2 NADH and the 2 ATP, because 2 ATP is what you have used. So this is actually the net yield after the glycolysis, after the complete oxidation of glucose during glycolysis. When I say complete oxidation, which means that the NADH is being produced. If you remember, when we were discussing about the incomplete oxidation, that time the production is less, because then the pyruvic acid is whatever is being generated is going through the decarboxylation steps, and it is going into the anaerobic oxidations and so on. So that's something different. So if you have a complete oxidation, you are going to generate 2 NADH molecules which are actually going to give you the 6 ATP at the end, and you are going to generate the 4 ATP, and 2 ATP you have utilized for channelizing the sugar molecules into the glycolysis, first by hexose, and the other one is the glucokinase. So let's move on to the next question. Okay, so we have one question from the Nisha. So where she is asking how to do physical restriction mapping. Okay, so I'm not very clear what she is trying to ask me, but uh, if you can elaborate what you mean by the physical restriction mapping, I could be very I could be it would be helpful for me to even give you the answer. uh, so far it is look like as you are asking something, but something is actually uh, incomplete. Means in what sense you are asking about physical restriction mapping, because uh, the if you can cite a example, or if you can say something about that, I think that may actually be uh, give me a better idea what you are trying to understand, so that I'll be able to answer those questions. So I request Nisha actually to elaborate what is mean by the physical restriction mapping, and I'll definitely be help you to understand that. Mm, if you see, I'm still not very sure, but I think what you're asking about is how the people are doing the restriction mapping. So for example, if I take the example of vector, for example, if I take example of puck 19, if I want to do a restriction mapping on, if I okay, so for example, if I have to get the complete restriction mapping of a particular DNA fragment, for example, suppose you have a 10 kb fragment. So what I am going to do is I am going to digest with the eco R1, and I am going to digest first of all, if you know the sequence, okay, then it is easy, then you can actually be able to do the physical restriction mapping simply by putting it into the software. And if the software will have the complete information of all the restriction maps for which we have the information about cutting sites, you can be able to do restriction mapping of that particular fragment. If you yeah, so that is what now I understood what you are talking, what we are trying to understand. So so there are two ways actually. One is you do it in the lab and try to understand what is the restriction mapping of that particular fragment. So suppose the fragment sequence is known to you, then you will be able to put it into the softwares. Like for example, if you do the NEB cutters, that actually will going to tell you all the restriction sites present on that particular DNA fragments. If you remember, when we were talking about the primer designing, that time we have, I think the Banesh has discussed about that, that how you can actually get the non-cutters. So uh, if you have a DNA fragments, just put it into the software, it will actually going to calculate, or it is going to analyze the whole sequence, and it is going to give you the positions at which the different restriction enzymes are going to cut. You can have the choices of choosing the restriction enzymes or which are going to cut single, or you can be able to choose the restriction enzymes which are going to cut not at all, and those are the restriction enzymes which are called as the non-cutters. Apart from that, if you don't have the softwares and you would like to do yourself, which actually people might be doing it around 30-40 years back, that you might have to digest the DNA on your own with the different restriction enzymes, then you are going to get, you have to resolve these fragments onto the high resolution acrylamide gels or high resolution agarose gels, and then once you got the different fragments, depending on the size of the fragments, you will be able to construct the restriction maps. I can give you an example. For example, if you have a 10 kb fragment and suppose you digested it with the EcoR1 and you are getting the 2 fragments or 3 fragments, depending on how many fragments you get, you can be able to tell that okay, these are the places where the EcoR1 is cutting and these are the site. For example, if restriction enzyme is giving you 3 fragments, this means it is going to have at least and suppose this is a linear DNA of 10 kb, then it is at least going to have the 2 sites. So depending on the size of those fragments, you can be able to very precisely predict where the EcoR1 sites are present on this particular fragment. But nowadays most of the people don't do this physical mapping by going through the experimental mode, because first of all it is going to be very time consuming. The second is you may not be able to very precisely get. So most of the people use the softwares which actually have the information about the cutting sites, and that actually will tell you the cutting sites or the cutting precisely the position where the restriction enzymes are going to cut. If it is not clear to you, you can still write to me and I will happy to help you and or explain you in much detail. I think we can take up the another until we don't have another questions. I think we can take up the some more questions from the assignments. So let's take another example or another question from the assignment. In the assignment 2, I have asked the question of the relationship between the blood glucose level and the activity of pyruvate kinase, and I have given the few observations and I have asked what are the observations are correct. And there was some technical problem when I was when probably we were editing this particular video part. So that's why there was some confusion, and we have rectified that later on. So what you can see is it is simple that when you have the high glucose level, the pyruvate kinase activity will go up, and the pyruvate kinase is present in 2 forms. One is dephosphorylated or the native form, or the phosphorylated form. So if you go to my lecture when I was discussing about the regulation of the glycolysis, I have discussed about the pyruvate kinase the regulation of the glycolysis through covalent modification, and I have taken an example of the pyruvate kinase. So the pyruvate kinase, when it is in the phosphorylated form, it is less active. When it is in the native form, it is more active. So as you can see, the option number 1 says that in the presence of high glucose in blood, the pyruvate kinase is more active. So that is correct actually. the second point is that in the presence of high glucose in blood, the pyruvate kinase is in dephosphorylated form. So, whether we write that pyruvate kinase is more active or whether we write that the pyruvate kinase is present in the dephosphorylated form, both of these statements are correct actually. Now, you go to the third statement. The third statement says that in the presence of low glucose in blood, the pyruvate kinase is more active. So, this is something which is wrong actually, because as I said, as you increase the blood glucose, the level of the activity of glycolysis will go up, and subsequently the pyruvate kinase activity will also go up. Now, let us go to the fourth option. The fourth statement says in the presence of low glucose in blood, the pyruvate kinase is in phosphorylated form. So, as I said, the phosphorylated form of pyruvate kinase is less active and the dephosphorylated form is more active. So, when you have the low glucose in blood, the pyruvate kinase is going to be less active, and that is why less active, or it is present in the phosphorylated form. That is why the statement 4 is also correct. And we have asked what are the statements are correct. So, the correct answer is the statement 1, 2 and 4 are correct. Now, if we have a few more questions, we do not have any questions so far. In the assignment 2 also, we have also asked at which wavelength the turbidity of the bacterial cell measured in the spectrophotometer to monitor the growth of bacterial cell, and we have given the few options. If you recall, when we were talking about the turbidometric method of estimating the bacterial cell growth using the spectrophotometer, what we said is that when you are measuring the turbidity, you are actually measuring not the absorbance, but the scattering of the light through the particles, because the bacteria is nothing but the suspended particle in the media. So, the bacteria will going to scatter the light, and because it scatter the light, that light will not reach to the detector, and as a result there will be some loss of light, and that actually you can measure, and the scattering is very pronounced. When you do measuring the scattering in the media, it is very pronounced when you are measuring it at 660 nanometer, and that is why the correct answer is 660 nanometer. Now, let us move to the next questions. So, this question also many of the students have asked the sir, how we are getting this and all that. So, this is very simple questions where we have in the assignment 2 we have asked how many bacteria were seeded or transferred to the culture tube in order to get 8.192 into 10 to the power 3 cells after 5 generations. So, what we are asking is that what is the number of cells we are inoculated into the media, so that you will get 8.192 into 10 to the power 3 cells after 5 generations. So, if you remember, when we were discussing about the bacterial kinetics, we said that if you have the one bacteria, or if you start with one bacteria after the first generation. So, if you see the generation 1, at generation 1 you are going to have the number of bacteria, that is 2. If you spend one more generation, it is going to be 4. If you go to third generation, it is going to be 8. If you go to the fourth generation, it is going to be 16. This means if you go with the this particular type of pattern, what you see is that after the n generation, the number of bacteria will be n equal to, because you started with the one number of bacteria, 1 into 2 to power n, where n is the number of generations. So, if you solve this, what you are going to get is the formula is log n equal to log n0 plus n log 2. So, if you use these formula and you would like to solve, what you are going to get, the answer is that if you start with 256 number of cells in 5 generations, you are going to get this number, which is 8.8192 or 8.192 into 10 to the power 3. So, if you calculate this, you will get the number of bacteria what is present or what is you are going to inoculate is 256, and that actually will going to give you this number. I strongly request all the students who are actually online to ask the questions as soon as possible, because now we only left with the 15-minute sessions more, and this is a great opportunity for you to sort out your queries. Otherwise, I am going to take up the question from the assignments which I have chosen. But if you have some questions from the assignment which you would like to discuss, this is the opportunity you can avail, and you can discuss these questions. So, let's take up the another questions from the assignment 3. So, many of the people have asked about these questions, where we have said that, so this is actually a question which is related to the genomic library preparations, and what we have asked is, a scientist want to isolate 3 kb X gene from a genomic library of mouse, which is actually 1.8 10 to the power 9 base pair. So, this is the size of the genome what we have given. If the library has these many number of clones, what will be the probability to get the desired gene fragments? So, if you see, this is the formula what you have to use, n equal to ln 1 minus p divided by ln 1 minus a by b. So, where the p is actually the probability factor. So, what you have to do is, if you put this values, for example, n equal to n equal to this, a means a is the size of the genome, okay, and the b is the size of the fragments. So, if you put this number, what you are going to, and if you solve this using the steps like the m1 minus p equal to n equal to this, if you solve this, what you are going to get is 1 minus p is equal to e to the power minus 0.583314, and if you simplify this, you are going to get 1 minus p is equal to 0.558, and that actually is going to give you a probability of 0.44. So, this is the option which is actually the correct options. So, if you go through with this, you will be able to solve this, and you will be able to get the correct answer. Let's move on to the next question. So, the next question is about calculating the melting temperature for a primer, which is actually the option which is the sequence is given. So, if you would like to solve these questions, you have to calculate, you have to use the formula of calculating the melting temperature for a primer which is above to 14 nucleotides. Okay. So, this is the formula what you have to use, 64.9 plus 41 degree into number of GC and C in the primer minus 16.4 divided by n, where n is the number of nucleotide in the primer. So, what you have to do is, you have to calculate the number of nucleotides. So, number of nucleotide present in this particular sequence, you have to count and put it into this formula, and that actually will give you a number which is 70.3. This is a very simple thing what you can do actually. So, the number of G and C what you have to first count. So, if you count 1, 2, 3, 4, 5, 6, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So the number of G and C in this particular fragment is 20, and if you see this whole sequence, the whole sequence also you can count, and that actually will going to give you the information about the melting temperature, which is going to be 70.3. Many of the students might have made a mistake when they were calculating the Tm. So for Tm calculation we have given the two formula. When first formula, which is actually been applicable for calculating the Tm when the number of nucleotide present in a sequence less than 13, and the other formula is which is actually been more applicable when you have the number of nucleotide more than 14. So you have to use the correct formula as per the length. So this total length of this particular fragment is somewhere around 23. I have not counted, but it's something like that. So that's the way you have to calculate this particular question. We have another 10 minutes left to ask me the questions, otherwise this session is going to be over. There is no technical issue, right? Okay, so let's I have taken another question from the assignment 4, which is actually the previous assignment, and what we have asked is which form of the plasmid DNA travels furthest when analyzed on the agarose gel, and we have given 4 options. One is open circular DNA, supercoiled DNA, covalently closed circular DNA, and linear DNA. So you know that when you are running the DNA on a agarose, the DNA molecules are going through the pores of the agarose particles, and that's how they are actually experiencing the friction. So when you are running a molecule into a into any supportive system like agarose or acrylamide or something, you are actually experiencing the friction forces on one side, and you are actually experiencing the electromotive force on the other side. These friction forces are actually governed by the size of that particular molecule, because the frictional forces are calculated like this. Okay, so this is the formula for calculating the frictional forces, where what you see is that the R is related to size of the particular object. So the bigger the size or the bigger the ball what you have, it is going to have the more friction, and that's why its velocity, because when the both forces are going to be equalized, it is going to stop making any absolute migration, then it is going to have a relative migrations. So the V is actually inversely proportional to the R. So bigger the R, bigger the lesser the length it is going to run on the gel, whether it is the polyacrylamide gel or to the agarose gel. So what you see is that open circular DNA, I am assuming that all, and you can imagine that all these are of the same plasmids. So if I have open circular DNA, supercoiled DNA, covalently closed circular DNA, and linear DNA, the linear DNA is going to have the largest R, because it is going to be present in the extended conformation. Compared to that, you are going to have the lesser area or lesser diameter or radius for the open circular DNA. So this is like for linear DNA, then you are going to have the closed circular DNA, the open circular DNA, then you are going to have the closed circular DNA, which is actually the this or this, but the DNA which is actually going to be present in a supercoiled form. So, you can imagine that I have the same ball which I have actually now made a supercoiled. So, this guy, the supercoiled DNA, is going to have the least radius in terms of the radius, and that is why this guy, the supercoiled DNA, is going to experience the least friction, which means it is going to run the very fast. So, that is how actually it is going to have the most migratory power, and that is why the supercoiled DNA will run in the front, followed by the covalently closed DNA, followed by the open circular DNA, followed by the linear DNA, and this is all in relationship to their surface area, which is actually indirectly related to the radius or the size in which they are going to be packed. So, now we have another question from the Nisha. What she is asking is that she has purified the two DNA fragment. One is 0.4 kb, the other one is a 0.9 kb, which were generated by BAMH1 digestion of recombinant DNA plasmid. Okay. After 30 minutes and after 9-hour incubation with DNA ligase, there are so many bands instead of 1.3 kb. Why are there so many bands in the original ligation mixture? Now, this is actually very simple. What you have is two DNA fragments which you have actually isolated from a plasmid. Suppose I am assuming that we have a single plasmid which is being digested with BAMH1, and you are getting two fragments, one is 0.4 kb, another one is 0.9 kb, or suppose you have taken the 0.4 and 0.9 from the two separate plasmids, irrespective of the conditions. When you have the 0.4 and 0.9, and suppose you are only digesting the DNA with a single restriction enzyme, then you are actually having the various combinations in which these two fragments are going to be combined. For example, the 0.4, because you can imagine that, so let me explain you in detail. Let us go to the PowerPoint and I will explain you. Okay. So, you have two fragments, one is 0.4 kb, the other one is 0.9 kb. Okay. Now, you can imagine that both these fragments are having the cohesive ends of the same BAMH1. Okay. So, you have the BAMH1 side on both the ends. Now, what are the various combinations in which the 0.4 kb and 0.9 kb are? So, suppose you have taken the 0.4 kb and 0.9 kb and put it for the ligation. So, if I put it for ligation, what will happen? You have, these are the combinations. You have all these combinations possible. You have this, and you can think of all these combinations like so actually because you are actually using the same dissection enzyme, there are many combinations are possible, because what you are using is one dissection enzyme, and uh, if you, so 0.4 and 0.4 can stick to each other, that will give you 0.8 kb. 0.9 and 0.9 can stick to each other because both have generated the cohesive ends with the same enzyme. So, you can get 1.8. 0.4 and 0.9, that is what you are hopeful that you will get it, but that will give you 1.3. But there is a least probability of getting this because the 0.4 and 0.4 is a very small fragments, and the ligation of 0.4 and 0.4 is much more favorable compared to that 0.4 and 0.9 is going to stick to each other, because the smaller the fragment, it is going to have the larger movement within the molecule and larger probability that these two fragments are going to attract to each other much faster rate compared to that 0.9, 0.9. So, there is a very least probability that you may get 1.8 base pair, but there is a high probability that you may get 0.4, 0.4 or the combination of 0.4 and 0.4. So, because of that, what will happen is at the end the 0.4 is going to be consumed in your reactions, and ultimately you are not going to get your desired product, that is 1.3 kb. For any such kind of applications, what you are supposed to do when you are using a single restriction enzymes, you always have to generate the you also have to make sure that both the fragments should not have the phosphate, the 5' phosphate, and that is why you might have to treat one of the fragment with the SIP, for example, the carcthymus, the alkaline phosphatase. So, if you treat one of the fragment with the SIP, then that particular fragment are not going to have, will not go through with the self ligations, which means if suppose I SIP the 0.4 fragment, then 0.4 is not going to go with the self ligation, which means the 0.4 will not going to go with the ligation with 0.4. It's only going to go with the ligation when the 0.9 will going to come and actually facilitate the phosphate. So, if you SIP one of the fragment, there is a highly chances that you may get 1.3 kb base pair. I hope I might have answered your questions. So, this actually, if you go with this kind of thing, you may actually be getting a ladder kind of thing, which actually be I do not know, I have not seen the the gel picture, but I predict that it might be having a ladder kind of situation where you might be getting the different bands with a difference of 0.4 kb, or some places you might be getting a 0.9 as well along with that particular thing, because what you are trying to do is you are actually trying to generate a polymer of 0.4 kb, because the probability of 0.4 making a ligation with 0.4 is higher compared to that the ligation of 0.9 to 0.9, because the 0.9 is going to give you the bigger fragments. I hope it is clear to you now. If you have any more doubt or questions, you can ask me again. I think we will go few more questions from the assignment, and then we will finish up this particular discussion sessions. So, I think a few students have also asked me about the difference between the linker as well as the adapter molecules. So, you can see that the linker molecule and under what conditions you are going to use the linker molecules. The linker molecule you will use in those conditions when the foreign DNA is actually a blunt end DNA. Okay. So, in those cases what you will prefer is you will put the linker molecules with the help of the T4 DNA ligase, and the linker molecule are going to have the cohesive ends. So, it could be cohesive ends for a same enzyme, like as Nisha was saying that the BAMH one, or it could be or different enzymes. So what you are going to do is you are going to take the foreign DNA, add it for the ligation reaction in the presence of T4DNA ligase, and then it is going to, if you treat this particular complex with the your recession enzyme, for example in this case it is the EcoR1, it is going to generate the cohesivants, and then when you put it for the ligation reactions you are going to get the recombinant DNA along, because the vector is going to be digested and the ligation, you will get the ligation product. Compared to the linker molecules, the adapter molecules are already been having the cohesivants. Okay, so you don't need to do a restriction digestion. That is the one first and the more important difference between the linker molecule and the adapter molecule is that for a linker molecule you might have to do a restriction digestion, then only it is going to have the cohesivants, whereas the adapter molecules are going to have the predetermined cohesivants. So what you are going to do, you are going to add the T4ligase and ATP, and it is going to generate the, it was going to attach the linker molecule on both sides of your foreign DNA, and then once the adapter molecule is attached, afterwards the species what you are going to get is same as what you are going to get in the linker molecule as well. And then you actually going to do a simple ligation reactions with the vector, which you also have digested with the same set of the restriction enzymes, and it is also going to give you the recombinant DNA. I think we have already crossed the 1 hour sessions, and I think the many of the students are not asking the questions. So I will wait for another 1 minute or so in case you have any questions. Otherwise, we will wind up the sessions, and we will see you in the next live sessions. So I request all the students to post the questions, because the session is going to be complete very soon, and you should not miss this opportunity to ask me your doubts or your questions. I have already taken few questions from the assignment which I thought are very important in terms of to discuss, and all those on those questions the student have asked me very lot of queries when in the discussion board. If we have any no more queries, I think I would like to wind up this live sessions, and I hope you might be enjoying the course, and if you have any more queries after this also, you can ask me in the discussion forum, and I'll be happy to answer. Thank you.